Welcome to the topic of electrophoresis. Electrophoresis is the separation of charged particles on the basis of their charge and also the molecular mass by the application of an external electric field. All the positively charged particles move towards the negative electrodes and all the negatively charged particles move towards the positive electrode. You have already studied that opposite charges attract while similar charges repel. This is the principle behind it. For the movement of the particles, there should be a fluid medium. Often it is a gel because it is fluid in nature, and since the particles we are going to separate are organic in nature, often in many of the biochemical situations, we use a buffer also because pH should not change during the entire process. And for the power supply we often use a battery. The electrophoretic apparatus is ready-made available and it is a convenient process for the separation of mixture of components. And there are some cases when the mixture may have particles with different charges at different places. In that case, we apply some chemical substances, we use some chemical substances which will provide all the molecules a uniform charge. For example, SDS, sodium rhodesyl sulfate. In that case, all molecules are having the same charges. All of them move towards the same electrode, oppositely charged electrodes, and they are separated on the basis of their molecular mass. Always, the smaller molecules are faster. So what does it mean? When the electrophoresis is going on, the smaller molecules move faster and will reach the other end first, while the largest of the molecule will be the slowest and they will move to the minimum distance. That means there is an arrangement of molecules on the basis of the size. Given here is the figure of a fragment of DNA, double-stranded DNA. Look at the picture in detail. You have studied it earlier. DNA has a charge. Look at the phosphate group. Each phosphate group has a negative charge. So in which direction will the DNA fragments move in an electrophoretic apparatus? Since the DNA is negatively charged, all of them move towards the positive electrode. Then if there are many fragments of such DNA molecules, how will they be separated? Not on the basis of charge, but on the basis of mass. The smallest of them will be the fastest. And what is the significance of separation of DNA fragments? For example, you might have heard of DNA fingerprinting. In DNA fingerprinting, we need to separate many fragments of DNA using electrophoretic apparatus. This is the technique to be used. Paper electrophoresis is a very simple type of electrophoresis used for the separation of small molecules and ions. For example, amino acids, small proteins, etc. Here we are using a filter paper. You can see in the figure, already the filter paper is moistened with the buffer solution, and the buffer is added to the anode chamber and the cathode chamber, and the ends of the filter paper are touching this buffer solutions in the chambers. And can you guess by looking at the figure in which direction are the ions moving? towards the cathode or towards the anode. And from that, you may be able to predict the charge of the moving particles also. Gel electrophoresis. In this experiment, a gel slab is used, and the gel slab is in contact with the electrolyte solution by means of a cloth wick. You can see it in the picture. There is the buffer solution in that one. Whenever we are using gels, it is more efficient. Therefore, even larger molecules can be separated. For example, amino acids and DNA molecules, RNA molecules, large proteins, etc. can be separated using gel electrophoresis. Polyacrylamide is a type of gel commonly used. Agarose is another type of gel commonly used for this purpose. In this picture, you can see the sample wells are nearby the negative electrode. From there, the samples will move towards the positive electrodes because the samples also might be negatively charged. And the smallest of the charged particle will reach the positive electrode earliest, while the largest of them will be the slowest. This is a very good method for the separation of charged particles. The same picture is given here once again, but here, what do you see here? Various bands. This is the observation we make at the end of the process. You see, actually here is the well. We applied the samples in these wells. From there, the fragments moved in this direction. So these are the smaller fragments, and this is the largest of the fragments. Okay. What are the different types of gels? You can see here agarose is a gel, while polyacrylamide is another gel, and both these are commonly used in the laboratory. You can see their comparison. Of course, in different situations, we select one of them. Here is the picture of a gel electrophoresis in which polyacrylamide gel is being used. You can see in the figure the long network of the gel. They are porous in nature, which means that many spaces are there in that gel. You can see a mixture of proteins is being applied at one end. Look at the different size of the proteins. Some of the proteins are smaller, while the others are larger. Some are of intermediate size. So based on the size, they are being separated. Look at the figure on the right side. The smallest of them have already reached the other end of the gel slab, while the medium-sized green colored ones are in the middle, while the large-sized orange ones are near to the beginning point itself. This is a very clear picture showing the principle of electrophoresis. And remember, polyacrylamide is one of the best gels used for electrophoretic separation of different sized particles. You see, what is PHOSPHATE and how is it prepared, etc. PHOSPHATE is actually polyacrylamide gel electrophoresis. Here, polyacrylamide gel structure held together by covalent crosslinks. They are tougher than agarose gel. So there are many advantages for using this polyacrylamide gel electrophoresis. And what are the applications of electrophoresis? Anywhere in the laboratory where molecules are to be separated on the basis of charge or molecular mass, we can use this. Estimation of the DNA, then analysis of the PCR product, separation of the restricted genomic DNA and RNA fragments, and confirmation of the newly isolated DNA, separation of most small fragments of DNA in forensic science, determination of molecular weight, and so many more applications are there in for electrophoresis in the laboratory. Actually, electrophoretic separation of molecules is one of the basic steps in any research methods. And again, applications of electrophoresis taken from another textbook. This is actually given in more detail.